Hi, in this video I'm gonna show you how to use this widefield fluorescence microscope. To get started, you want to turn everything on. First, you're gonna turn on the computer by clicking on this button. Then you're gonna turn on the monitor. Printer, you don't have to turn on. Next, you're gonna turn on the chiller. The chiller is over here. After you turn on the chiller, you have to set the temperature of the chiller to 25 degrees. To set the temperature, you need to click on output temperature until the setting, the reading changed to set temp. Right now, you can see the temperature is set to 30 degrees. To change the temperature to 25 degrees, you want to hit this button until the set temperature is 25. The response is kind of slow, right? So you have to hit and wait until you see a change of your action. Alright, so now the set temperature is set to 25 degrees, and now you want to hit enter. To hit enter, you have to again hit this button until the reading set manifold. Right? Manifold is the current temperature of the chiller. You will see that this number will go up, so always have like a look on this number. Make sure the temperature is set to really 25. So next, we have to turn on the compressor, and the compressor is creating air for the air table. So to turn on the compressor, you have to hit this button. You can hear the sound, right? This is the sound of the compressor. It's not comfortable to hear it all day, but what I can tell you is this will stop after the compressor reach a certain pressure. And here and then the compressor will jump on. It's happened every 25 to 30 minutes, so you don't have to be worried to suddenly hear a sound, right? A loud sound coming from the compressor. Now let's turn on as well one of the most important part of the microscope, which is the laser. Right? The lasers can be seen over here. And we as well need to turn on the camera, right? So to do that, we gonna have to hit this button over here, and this gonna turn on both the camera and laser. So let's do it together. So let's check whether the laser is on, right? So let's look at the laser, and as you can see over here, you can see the reflection of the green laser light, right? So which show that we have successfully turned on the laser. And the program to control the microscope is Andor. As you can see here, Andor Solus 64-bit. This program is used to start our measurement, to stop the measurement, and do everything we need for the experiment. To turn on the program, double click with the left mouse on the icon, and then you will see this symbol is coming up. Now let's wait about 10 seconds, and here you can see this is the interface of our program. For now, you don't have to do anything. The program will cool down the camera. As you can see here, this is the current temperature of the camera. Right now it's at 10 degrees. We have to wait until this reach the temperature we want. So the camera is cooled down to minus 60 degrees, right? And this happened automatically, so you don't have to do anything, right? So we have to wait until this became blue before we are able to do any measurement, right? So that's why we have to turn on this program first. Alright, so now we're gonna get started and do our first measurement together. Just briefly show you the microscope. So the microscope consists of a light source. This is our laser. Over here you can see the neutral density filter. This filter is used to vary with the laser power. So based on how you should move it, you will see that the laser power getting through is getting weaker, or getting stronger. So if you move this neutral density in this direction, you will see that the laser power is getting higher. And if you move then in this direction, you then can lower the laser power, right? So the neutral density filter is used to block a certain amount of laser, and we are using that to control and maintain the laser power for our measurement. So after the laser goes through, right, it will follow this path, right? It will go along this pathway, right? Here are a couple of lenses, mirrors, and so on, and then this will bounce back, bounce here, and then go this way, and then after like a couple of optics, it will reach a very important mirror. This is the so-called dichroic mirror inside of this cube, and then the dichroic mirror will allow the laser light to bounce up. And now let me open this for you to see. This is a very important piece of equipment for our microscope, and this is our sample stick. And the laser will go, right, again coming from this way, bounce on this dichroic mirror, will be reflected up, and then it will actually be focused on the aperture, right, of this objective lens, and then the laser light will later on be focused, right, on our sample through this objective lens, and laser light will hit our sample. It will excite the dye, right, dispensing the sample, and we then can collect the emission, right, fluorescence emission of the dye molecules coming, dispensing our sample using the same objective lens, and then the fluorescence emission will be, right, directed down, right, this way, and then now the fluorescence emission gonna hit the dichroic mirror, which is inside of this cube, and this time the dichroic mirror will let the fluorescence emission going through, right? This is a really cool mirror we have. It's allowed light to be reflected or transmitted based on what wavelength we have, and for a longer wavelength we let light going through be transmitted. Why? Because we need to have our fluorescence emission hitting the camera, right? The camera is our detector. So now I will show you how it can go to the camera. Let's open this thing. So as you can see, right, we're having another mirror sitting over here, right? So light will this time go through the dichroic mirror, hit this mirror, and then it will be reflected, and you can see here, right, here's another tube and a lens inside, and then this time, right, light will be going through, right, enter this tubing, and eventually, right, it's gonna be, right, it's gonna hit the opening of this camera, and then we are collecting, right, light of the fluorescence molecules in our sample using this camera, and then the signal will be shown here on the program. And now I'm gonna show you how to do the first couple of measurements. The first thing we have to do is we have to put on a sample, right? And I'm gonna show you how to do that based on a special we have. The sample consists of fluorescent spheres, right? 1 micron in diameter, and the sample is really bright, right? So this is a really good sample to get started, to learn how to focus and learn how to operate it, right? So let me show you how to do that. So first of all, right, we have to remove this cover, right? The cover is used to actually control the measurement temperature, right? If we want to. For now, we actually don't have to do it. And as well, this cover is used to maintain the environment for of a sample, right? So we can do measurement at, for example, different humidity, right? If we want to. Again, this is not the part of this training. So now I'm gonna show you how to do the measurement just in air, right? At room temperature, right? So nothing fancy, just to get started. The first thing we have to do is we have to put a small drop of oil, right, on the objective lens. And oil is a special oil. It's called immersion oil, right? So it has the same refractive index as our glass cover slip, right? We are preparing all sample on a glass cover slip, right? So immersion oil can be found in this small transparent box. You can see over here on the shelf in this box, right? So I'm gonna grab it and then I'm gonna show you how to put a drop of oil on the objective lens. You can see there's a small bottle inside, the small bottle with a white cap. This is our immersion liquid, right? Or immersion oil, right? So now we're gonna put a very, very small drop of this oil on the objective lens, right? Let me show you how to do it. So I'm gonna take, right, this out and then check whether, and then remove as much oil as possible, which is, you know, initially quite a lot, until you see there's only a little bit of oil left on this thing, right, on this plastic applicator, right? And then now we're gonna wait a little bit until a small drop of oil is formed at the tip, and now you're gonna go down and carefully touch the surface of our objective lens, right, with your plastic applicator. We shouldn't put too much oil, otherwise oil will just run down the objective lens and damage the optics of our microscope. So it's better to put too little in the beginning, and then if we see this is not enough, we can go back and put more. Now it's time for us to put on our first sample I was talking about. Is as well sitting on the shelf. We want to take out one sample which is intact. A couple of them are a bit cracked, right? So take out one sample, and usually the good sample is sitting on the top, right? So you don't have to dig any sample which is a bit more below. And then, right, so this looks like that. You can see it said, right, 2 micron spheres, right? Those are the so-called polystyrene spheres, which spheres were stained with a lot of dye, right? And the dye has the correct wavelength, right, for our laser, right? So we gonna use it to calibrate or to train you on the microscope. To take out the sample, right, you open the lid, right, and then you can actually use your finger, right? Always grab all your sample at the side, right, at the edge of the of this glass cover slip, and right, lift it up like that, and that's why check the sample, check whether the, you know, whether the top is top and bottom is bottom, right? Bottom, when you look at bottom, you should not be able to see a film, right, deposit on the glass cover slip. If you look on the top, you actually can see, there's actually something sitting on the glass cover slip, right? So we want to make sure we are putting all our sample in the correct way, right? So top of the sample should be pointing up, right? So make sure you are doing that correctly. So now I'm gonna put this sample on the microscope. So I'm gonna put it down like that, and one thing I want you to do is make sure the sample is centered, right? So do not tilt it, make sure it's really balanced, right? Make sure it's sitting stable, right, on the stage. You can see right now it's pretty well centered, right? You can see here, right, this distance, right, is about the same as this distance, right? Alright, so I always put my sample horizontally. This is just convention, right? So we know later on, if we have to discuss the direction of anything, we know how this actually was aligned on the stage. Perfect. So now, right, we are almost ready to start our first measurement. Now let's try to change the laser power, right? As I have shown you, we're gonna control the laser power using the so-called neutral density filter, right? The polystyrene spheres are super bright, which means they have a lot of dye, right, attached to the spheres. So we want to use a laser power as small as possible, right? So which means our mission is right now, right, to move, right? You can put your finger in there and you can see, right, that the laser is right hitting my finger. And now our mission is to change, right, to lower this laser power a little bit by moving the new filter down, right? If you move it down, right, you can see, right, that the laser is now hitting the darker area of this filter, right? Darker area means this area gonna absorb more laser light. Whatever is coming through is now weaker, right, in intensity. So let's try to move everything down as much as possible, and then, right, you can see the laser power is now much weaker. By lifting up this piece of equipment, right, which is actually used to block the laser, I call it a shutter, right? A shutter is used to shut off the laser. So if you lift up this shutter like that, you then will see, right, now the laser is, right, is now being focused on our sample, and what you can see is, oh, the laser power we currently have set is still too high, because you can see, right, the fluorescence even with our eye. You can see this light is kind of yellowish greenish, right? And if you compare that to the laser, you can see it's more greenish. So we know that, oh, what we actually see right now coming from the sample is actually already our fluorescence emission, and we can actually see it with our eyes, which means it's a little bit, right now the laser power is still a bit too strong. So let me as well set this, change the laser power a little bit more, right? So I'm gonna actually grab right now the filter directly, and then I'm gonna move it manually, right? I'm gonna, as you can see, right, my fingers really touching the filter, and now I'm gonna move it so that the laser is going to an even darker area of this neutral density filter, which means more light should be blocked, right? Right, let me do it as much as possible, right? And then now, right, you can go back and compare. Oh yeah, what I can see now is this light, right, coming off the sample is much dimmer. So now we are ready to do our first couple of measurement on this wide field fluorescence microscope. Alright, so now let's go to the computer, right? And then let's look at a couple of settings. Alright, so this is still our Andor Solid program, and we are using, as I said, this program to control all our measurement on the microscope. And right, so before we do a measurement, we have to know what are actually the imaging parameters. How are the fluorescence videos collected? To do that, we have to check the parameters by clicking on this acquisition setup symbol. Right? After I click on that acquisition setup symbol, you can see which window will pop up. You can see right now, right, what we are doing is, right, for example, the exposure time, right, is currently set to 0.1 second. Each fluorescence video consists of a series, right, of images, right, for example 1000 images. We also call those images frame, right, 1000 frame. And this exposure time is the time it took to take one image, right, so exposure time. 0.1 is usually our, right, the time we are using to record those videos is just a standard parameter, so you don't have to do any changes. Next important parameter is so-called kinetic series length. This is now the number of images, right, we are collecting to obtain one fluorescence video. Again, 100 is again a default setting, right? We can later on, if we want, we can change that to 1000, 10,000. It's up to us, right? But for now, again, this is fine. So important is as well here, right? Here you can see our gain setting, right? You know, gain should be as much as possible, right? So always, again, default is 3. And now another important setting is here, output amplifier. It is very, very important that initially when you are imaging the fluorescent spheres, that this setting is at conventional. Don't set this to electron multiplying. You have to carefully check that before you start recording any data. Conventional means standard setting. Conventional setting is used for bright sample, like the fluorescent spheres we have, right? Don't turn on electron multiplying, otherwise you are risking to damage your camera, right? So please, it's very important that you do that. Alright, so another thing I want to show you is binning, right? Means binning or region of interest, ROI, right? This is showing how big, right, is our videos, right? Right, so here you can see default settings is 128 times 128 pixel is the width, right? Width and height of our image, right? And 1 pixel corresponds to 200 nanometers. One image reflects about less than 10 by 10 micron area of our sample, right? So we're really zooming in, right? Looking at some really local region. What's going on, right? How does the dye move within those polymer region? And binning, right, is actually used to enhance the signal, right? Again, 2x2 is standard, so you don't have to do any changes for now, right? Alright, so this is all what I want to show you, right? So confirm that this really conventional setting, right, before you do anything, right? And as well confirm exposure time is 0.1, confirm that kinetic series length is 100 frame, right, video we're gonna collect now, right? And then next click on OK. And before you gonna record one image, you gonna have to do a couple things. So usually we have to turn off light in the room before we start recording any data, right? Because light is interfering with your actual measurement, right? But for the purpose of this training, I will not turn off light, right? Alright, so that you can actually see what's going on. However, I'm gonna put on a cover, right, for the microscope, so that we are reducing the amount of light hitting our sample. And this light is actually the background light. Now, right, we then can start our very first measurement. And I will show you how to do that using Andor, the program, to control the microscope. We first have to click on the fourth button from the left. It said take video if you put your mouse on it, right? Take video means the camera is running, right? You can see, right, what's going on in your sample, but you are not recording any data, right? In contrast to the other button which said take signal. Take video means the cameras keep running, right? So we are using take video mode to actually doing some adjustment. And then after you feel you are ready, right, to collect your first data set, then you're gonna click on take signal to get a video at the end, right? And which can be later on, right, saved, analyzed, and so on, right? All right, so for now, right, we just want to see what's going on. So I will click on take video for now, right? And this is what we see, right? All right, so now next thing you have to do is you need to open the shutter, right? The shutter is over here. Without opening the shutter, obviously the laser light won't hit your sample and you won't be able to see any signal, right? So what we're gonna do now is we're gonna move this shutter to the side, and now, right, you will see, right, always check whether the laser light is hitting your sample, right? Yeah, and indeed you can see the laser hit the sample, and you can even see some fluorescence using your eye, right? This is a good sign. You're gonna see, right, that actually an image like that is shown by the camera, and this is still not what we want to see. And the reason why is the focus is currently bad, right? So I'm gonna show you how next to do the focusing. So the focusing is done, right, using this knob, right? So there's like a fine adjustment here, and as well you have a coarse adjustment here, right? So always use the fine adjustment, right? So to focus, you either have to turn this knob clockwise so that the objective lens is moving up, or you can as well turn this knob counterclockwise, and then you're gonna lower the objective lens. Why? Because as I mentioned, we know that the focus is bad, means either the laser is now hitting somewhere above the sample or somewhere below the sample, right? So our mission is to have the laser light focus perfectly on the sample, sitting on top of the cover slip, right? So now what you have to do is you have to turn the knob, and at the same time looking at your video, right, and try to see whether you're getting a better image, right? So let me do it for you. So I'm not the key about focusing is going slowly, right? Slowly until you get what you want, and as well don't keep going into one direction. If you keep turning for example clockwise, then you might be going the wrong direction, right? So the idea of focusing is actually going clockwise a little bit and then counterclockwise a little bit until you get the best results. So I have adjusted the focus, right? I went back and forth, right? I turned this knob clockwise and then counterclockwise, and I did it for a while until I obtained this image. This is a fluorescent image, right, of our polystyrene fluorescent spheres. You can see, right, the round spheres, right, over there, and the diameter of those, right, circles is about 2 micron, right, as we would expect it for this kind of spheres. All right, yeah. So this is, right, the first image we have collected together, right, and your goal, right, as a new student in my lab is to be able to put on a sample and then focus, right, good as possible, so that you are obtaining an image I just showed for the fluorescence. And the idea is you want to get an image as sharp as possible. So if you see that the image is blurry, right, it's indicate that your focusing was not perfect. So now, right, I'm gonna show you for some next how to put on your actual sample and how to take off the fluorescent sphere sample and so on. So before we do anything, we need to, of course, stop. And how to stop that is by clicking on this red abort acquisition. Simple. To change the sample, what I'm gonna do is I'm gonna use a tweezer, right? And then I'm gonna grab the sample, remove some oil right on the bottom of the sample, and then put it back into our sample box. When you are done with the measurement as well, make sure that the shutter is pushed back, right? And then as well make sure that the laser is not hitting your sample anymore, that the laser is not hitting the sample when you are removing the sample. You remove the sample by grabbing your sample using a tweezer, right, like that. And I'm using the one of my finger, right, as well to hold, right, because sometimes you push the sample back when you are trying to grab it. So with another finger you try to stop the sample from moving around, right, and then now you grab the sample, and then next thing you do is you're gonna drop the sample, right, on a clean piece of Khanh-Hoa, and then now you're removing excess amount of oil, right, on the back side of the sample, like that. And then now we have to put the sample back into our box, right, like that. And then make sure you put this sample back into the box and put it away. We're done with this. So now I'm gonna show you how to put on a real polymer sample, right, which was doped with some dye. For the demonstration, I'm gonna put on a simple polyethylene oxide film doped with some SRB dye, right, sulforamine B dye, and then we're gonna quickly record one image in one video, and then at the end I'm gonna show you how to shut everything down. So now, right, before you put on your real sample, right, make sure you're re-adding some oil, right, back on the objective lens, because after each time you're putting a sample, right, some of the oil is actually removed by the sample. So now, right, I'm gonna added a little bit oil back, right, on the objective lens, right. So make sure you are removing excess amount of oil, right, from this applicator, right, and then you wait a little bit until a drop is formed at the tip, right, like that. And then now you can touch, right, the objective lens again with this applicator, right, and you check, right, whether you see a drop of oil, a small drop of oil is on the top of your objective lens, and I actually can see it. And we're gonna do it in the same way as before, right. So make sure, right, you know what is the top of your sample, and right, make sure you put the sample on in the correct way, right. Top is top and bottom is bottom, right. Don't put the sample upside down, otherwise you will not able to do the imaging. Here is the polymer sample we have. This is polyethylene oxide with dye, right, and then we will put this one on and try to image it again, right. So I can see, right, that right now I'm looking at the actual sample, right, on the top of a glass cover slip, right, and then here, right, is on the other side, this is the back side. So make sure, right, the polymer is pointing up, right, and then I'm gonna put the sample on, right, the microscope like that, and again horizontally, right? And make sure that is kind of centered, right, as much as possible, like that. Now, right, this is a dye sample, so we know that we don't expect to see a whole lot of fluorescence coming out of both samples. So we need to now increase the laser power, and as I have shown before, right, we're gonna do it by moving the neutral density filter up, so that the laser is going to an area which is less dark. It means more light can go through, and then less light will be absorbed. Right. So now, right, yeah, I mean, to me this looks fine, right? So I'm gonna increase laser power a little bit more, right? And this looks fine to me. This is pretty powerful now. And as well, we can double check by removing the shutter, right? And you can see that, the laser is pretty powerful. Alright, so now, right, we are good, right? So move back the shutter, right, if we don't need to do the measurement for now. And right, so now we're ready to image the sample. And so now we're gonna make sure you put the cover on the microscope, make sure, right, you turn off the light, right, on this. And now, right, I would check whether the focus is good, right? I need to focus, optimize my focus. So I gonna click on again the take video button first, and it's noisy like that because the shutter, right, is still there, right? So now I will remove the shutter, and still nothing's going on, right, because the focus is bad, right? So now, as I have shown before, I'm gonna turn the focusing knob until I see a good image. Alright, you can see, right, that I'm adjusting the focus right now. I adjust it until I see that there are fluorescence molecules, like that. Right? I see there's some fluorescent spot moving around, right? As you can see over there, right? This is probably one of the, like, the focus we can have. It's not the best possible focus, but and now, right, if you are interested to record a video, right, I will show you how to do it. So first you have to click on Abort Acquisition, and then you're gonna click on Take Signal, and you have to wait, right, until you hear another click, and this indicates that you have successfully recorded this video, right? And now you're gonna go to File, right, and gonna click on File and go to Save As, and then you can create a folder on your desktop, right, with your name, right, and then you can, I will call this student, and then you're gonna go and create another folder with the date of experiment, and so today is 06/27/2020, and then you can then give your file name, and so we know this is PEO, and then we can say this is PO1, right, for the first measurement we do, and click on Save. Right, after you save your file, right, and this is saved as a SIF file, you have to close your save file, right, in order to record a new video, right. To close the previous file, right, the save file, you click on the small X over here, right. So now, right, I'm gonna show you how to shut everything down. To shut everything down, right, you first can close your program, right. So close the Ender program, click on X, and then it's gonna ask you, do you wish to save acquisition setting, blah blah blah. You will say no, right? We don't want to change any acquisition setting. And then after that, obviously you're gonna log off, right, from the computer and so on, right? So do that. And next thing we do is turn off the laser, right, and the camera. We're gonna do that based on right here. Don't turn off the chiller for now, right? Let the chiller run for another 5 to 10 minutes, right, to further cool down the camera, because the camera is still hot from running, right? So let the chiller run for another 5 to 10 minutes. We're gonna turn it off after we clean up the microscope. But the compressor, right, now can be turned out. Alright, so now I'm gonna show you how to clean the microscope, right? To get started, we first gonna remove the sample. I'm gonna show you how to do that. So you're gonna use a tweezer and then use one finger to hold the sample from at one side, and then use tweezer to grab your sample. Lift it up like that. And if you plan on keeping the sample, right, you can again clean the sample. Right? Use a clean piece of Khanh-Hoa and then clean the back side of the sample like that. And then put it back into the box. Now I'm gonna show you, right, how to clean the objective lens before we shut down the microscope. The objective lens will be cleaned, right, using, right, again, this is like a white box, a white container, sorry, containing our immersion oil, right, and as well you can see there's some swab, right, and as well there's cleaning alcohol, right, and as well inside this container you can see there's some lens cleaning tissue like that. So now I'm gonna show you, right, how we're gonna clean the objective lens. You're gonna take a piece of swab, you're gonna take a piece of our lens tissue. This lens tissue is a special tissue, so please don't use Kim wipes or use paper towel. If we're running out of lens tissue, please let me know. This is a special, really fine and soft tissue for objective lenses, because objective lenses are very, you can get a scratch really easily and very expensive, right? So what we're gonna do is you're gonna put this lens tissue on the swab and then try to wrap it around like that. Right, like that, and then you're gonna add a few drops of methanol. Probably 2 drops will be fine. Add it on the tip of this lens cleaning tissue we just prepared. 1 drop and 2 drops, and you're gonna take this, right, and then clean, right, the top by going one time over the lens, objective lens, and then go another time like that. And then as well carefully remove any excess amount of oil you have, and if you can see it, some oil is running down somewhere, right, so clean the side of objective lens, and you're done. Now, right, you're gonna put everything back as how you found it. So put back this humidity box, right? We don't need it for now, that's why I didn't show you how to use it, but essentially it's just the same. It's not hot, right? So put it back like that, and now, right, we're ready to turn off the chiller, right? Turn off the chiller like that, and make sure you put everything back into the box and put everything away. So this is a brief demonstration how to use this fluorescence microscope. This is just a start, right? Good starting point. You need to get more training, right, to be more comfortable using it. And yeah, so thanks for watching.